Bismillahirrahmanirrahim. Welcome back, friends. Welcome back to another video lecture by Kami Microbiologist. Today in this video lecture we are going to talk about the blood smear examination, that what we examine in the blood smear. If you guys remember, we already talked about the blood smear, what is blood smear, how to make it, what we see in the blood smear, each and everything I already explained. It is present into my channel, so it was our first video. If you missed this, so it is present into my channel. I will give the link also, and you will also check in the I button as well, so then you will easily find that what is actually blood smear. So I already explained the introductory portion of the blood smear. So in this video we are just going to see that what is examination of the blood smear, means what we examine in the blood smear. So my name is Muhammad Kamil Khan and I am the microbiologist. Let's start the video. First of all, that what we are going to talk about in this video lecture. So we are going to see the red blood structure, we are going to see the different examination in blood smear, like different terminology: anisocytosis, spiculocytosis, hypochromia, hypochromic, polychromasia, etc. Before going to explain each and everything, first of all we have to know about the red blood cells. I already explained this red blood cell, but here I will just a little bit talk about that what is red blood cell. So again, it is the type of the blood cells which is mostly circulating into the bloodstream, and it contains hemoglobin, that is a protein, and this hemoglobin carry oxygen to from the lungs to all part of the body, and it also transport the carbon dioxide to all part of the body back to the lungs, and then it will expel out from the body. So this is all about the red blood cell function. Now then we have the lifespan of the red blood cell, that is about 120 days, and after 120 days the red blood cell are broken down and again recycled. Moreover, as we already talked, that it is mostly important in the circulation, means to provide oxygen or to transport oxygen to all part of the body. Let's see the structure of the RBC. So the RBC structure is biconcave in shape, biconcave disc like in shape. I hope you know the CD disc. So it is just like in this shape, okay, and its surface area to volume ratio is increased. Why? Because it is due to the absence of the nucleus, and it is due to the absence of the granules. And the shape of the RBC, which is very just like biconcave in shape, due to this way we can say that efficient gases exchanges can be occur, because the cell contain a lot of area for the oxygen and for the carbon dioxide. Then we have the diameter of the cells. So the diameter of the cell is 6 to 8 micron. The normal and mature RBC have 6 to 8 micron diameter, and the width is 2 micron. Then remember that this RBC red blood cell do not have nucleus, and they do not have organelles. They have just mitochondria. Moreover, due to the absence of organelles and nucleus, it can easily uptake a lot of hemoglobin, that is the protein, and due to this way a lot of oxygen can be capable, and it can carry a lot of oxygen due to this way. Now this is the exact structure of the RBC. As you can see, this is the RBC structure. This is just like a disc in shape, okay. So this is the membrane, and this is the cytoplasm containing the hemoglobin. Let's see the first terminology that we are going to see in the blood smear, that is about the anisocytosis. So what is anisocytosis? So anisocytosis means the variation, the changes, or the modification, or we can say the abnormality in the size of the RBC. We call it anisocytosis. Aniso means differences, variation, etc. Cytosis means cells. Then we have another point that says that, remember, the RBC, for example, means the normal mature RBC have normal lymphocytic nucleus size. Okay, and remember, whenever you want to see the anisocytosis and whenever you want to study the anisocytosis, so before that you have to know about the MCV. I already explained the MCV, and you also know about the RDW, and I also explained the RDW. You can go back to my channel and watch the video about the MCV and RDW. It's already explained. So MCV and RDW is very important to know about the anisocytosis, because in MCV we mostly talk the average RBC size and their volume, means the RBC size and the volume is actually called MCV. And RDW means a red cell distribution weight, in which we measure the variation of RBC amount in volume. Okay, so if we know the value and RDW and MCV value, so then it is really important in case of the anisocytosis. Another point says that, remember, if we see that MCV value get high, okay, for example it get high to 100 femtoliter, so it means that we have large cells, and the large cells are called as macrocytic cells. And when we see that the MCV value get low, so then it is called as microcytic, means the cell size are very small, and we call it microcytic cells. Then another point says that we always compare the RDW and MCV value, so both these values should be compared in case of the anisocytosis. So this is the size of the RBC, just similar to the size to the nucleus of the lymphocyte. This is the normal RBC as you are seeing in the smear. Now let's see the types of the anisocytosis. So we have first type, we call that macrocytic blood picture. So if you want to see that what is macrocytic blood picture, so macrocytic blood picture is the field in the microscope when you make this blood smear, and when you see the field, and at a particular field if you see that the cells are larger in size. Moreover, if you see in the blood report, if you see that MCV value is high, means higher than 100 femtoliter, so it means that this is called macrocytic blood picture. And in this case the RBC diameter will be 9 to 14 micron. Next we have another type of anisocytosis, we call it microcytic blood picture. This is opposite to the macrocytic, because in this case the MCV value will be less than 80 femtoliter, and in this case the RBC are very small in size, and their diameter is 5 micrometer. Now these are the structure of the RBC. As you can see, this is the normal cell structure, and this is the small cell structure which we call that macrocytic, and this is another RBC structure which is larger, we call that macrocytic. How you will differentiate macrocytic and macrocytic? So you have to know about the MCV. Remember, if the MCV value is high, so then the cells which you are looking in the field of the microscope, it will be macrocytic. And if you see the MCV value get low, so when the cells, when you are visualizing under the microscope, so this will be the microcytic. Okay, this is really important. Then we have macrocytic cell condition, means in which condition the cells are become larger, or we call that macrocytic cell. The first is vitamin B12 and folic acid deficiency. So due to vitamin B12 and folic acid deficiency, what happened, the cell are become larger in size. Liver disease is also another cause. Hypothyroidism means low level of hormone in the thyroid gland. Autoimmune disease, aplastic, pernicious and hemolytic anemia. These are the three types of the anemia due to which the cell we can say that the cell size are become larger. We have alcoholism, when the person are drinking alcohol, so in this case their RBC size are also larger. Splenectomy means the removal of spleen. Then we have increased erythropoiesis. When there is a large number of erythropoiesis, so obviously in this case we have macrocytic cell also. Then we have another condition due to which the macrocytic cell will be happen. So let's study the macrocytic cell condition. So it is due to the iron deficiency anemia, IDA. It is also due to the thalassemia, spherocytosis, in which the cells are spherical in shape. It is due to lead poisoning, sideroblastic anemia. So all this is about, okay. So in the next video we will explain hypochromic RBC or hyperchromia, each and everything in the next video. I hope this video is helpful for you. Thank you so much for watching.